First, what is GAP? We were started by a consortium of pharma companies and charities designed to take an orthogonal view of how to re-engineer clinical trials in Alzheimer's disease, and not an ambitious goal, make them faster, better, and more effective. We've been doing it. This is our 10th anniversary, and in the course of that, we've been involved in all but one of the phase 3 therapeutic studies, a number of phase 2s, and learned a lot, mostly of what we don't know. Hence, my discussion today will be on how to better characterize with more precision and with greater speed the folks we want to have in clinical trials. One last comment. All the good data you will be seeing comes from a colleague of mine by the name of Sam Lee, a young PhD who was actually funded by LifeArc at Leicester, and he's been great help. So I always come to these things and wonder, well, what am I really going to, what am I going to hear that is a little bit novel, and more importantly, so I'm going to start with just quickly saying what we see as a way to change the whole Alzheimer's research field, and especially for our hosts, LifeArc, as they drill down into rare Alzheimer's disease, with the caveat that I don't think the rare Alzheimer's diseases are that rare, and we'll go into those data. But if we are, if we're going to do a better job, we have to detect the multiple pathologies with higher sensitivity and specificity related to dementia. We have 5 we're focusing on. I'll come to that. We need to measure the level of the pathology relative to risk of illness and ease of interpretation, because if you're going to scale, average docs in the field need to be able to actually describe the situation to their patients and give a care pathway that makes some sense without a lot of complication. We really emphasize the need for speed. That is, whatever we're testing in the way of in the field for diagnostics or trials, we need a very fast cycle time. We're very enthusiastic about the blood-based biomarkers for that reason. And scalability. You're certainly not getting PET across the world. You are not going to get minus 80 degrees Fahrenheit, minus, what is that, about 26 degrees centigrade freezers to put blood into remote locations. And so thanks to LifeArc, we're testing in a new biomarker study the ability to use blood spot technology to acquire in almost any environment the ability to first get the blood sample and then second to test it. So here are my I picked a Canadian, by the way. The Gretzky predictions. 1, there'll be a blood marker for alpha-synuclein within 3 years. Could be faster than that. That's remarkably important because, as those of you that know the field, alpha-syn is both to Parkinson's and to Alzheimer's a really elusive protein, and it's elevated in Alzheimer's, and we don't really focus on it. But we know it's in the background and could be leading to the rare diseases my friend Giovanna is looking for. Second, I think we're going to see more than 3, but at least 3 MTBR BBMs for tau. And that's a huge area of focus. We've got a great marker, which we talked about in the identification of amyloid. Tau is the next one. Huge amount of work being done in that area. And what we're always looking for is what will scale, what has a short cycle time, and cost. And last but not least, we need to understand that the field broadly, rare diseases and the more general Alzheimer's, we're trying to push to the cognitively normal, pre-symptomatic. We've got to measure them differently. We do not have a blood test that's ready for that cohort. We do not have digital cognitive tests for that cohort either. But that is where the research is going to go, and a lot of effort being put there. So I just want to take you quickly back. It was in London, 2017, 9 years ago, and at AAIC, just a few miles from here, we showed for the first time a battery of blood tests from 4 different groups that showed great sensitivity and specificity for amyloid and some other proteins associated with Alzheimer's disease. Great. We owe a lot to WashU. I would say that of the 4, and the day of its arrival, the WashU C2N mass spec-based 4240 was a game changer. And it created the whole environment for, well, we can go after this. We can find these plaques, and we can find them in a way that will allow for more enhanced research. The rest of these companies were right behind it. Fujiray Bio, though, jumped ahead. So I wanted you all to see, and I apologize for the American focus, but the FDA is awash in blood test applications right now in this general area. This graph generally shows you who's already got a clearance. That would be Fujiray, Lumipulse. Very important. It's a p-tau 217, which is a remarkable range of measurement. It's almost a miracle based on people that aren't me, because I'm not clinical or a scientist. The ability, sensitivity, and specificity of that particular test for amyloid plaque, and in certain versions of it, it can also show a reasonable correlation for tau. Then you see our friends from Roche did something so clever, back to scale. When they went and applied for this knockout test, this is a test that has a 0.9 or better correlation with negativity. The negative predictive value of this test is quite high. But it was also had a label that read, for the use by primary care in the diagnosis and treatment of Alzheimer's patients. So in America, and I don't think it's that different in UK, the front line of treatment are primary care physicians, and they didn't know if they had a test they could interpret. This thing is simple, one cutoff. You're either in or you're out, and if you're not negative, now the docs know that they have to start working you up for the possibility of having some form of dementia, particularly Alzheimer's. And you can see the rest of these names. Now look at this slide. This is what we know is in front of the FDA or in the marketplace coming forward. I showed you where we were in 2017. Here we are 9 years later, and this is a lot of companies, and this is a lot of tests. And there are 15,000 different proteins being taken on here. Is probably an understatement by an order of magnitude, but proteomics is a tricky business. And these folks will report SomaLogic, who's great, so's Olink, so's Seer, but they will report these huge number of proteins, and I can never tell, because I'm just a layman, how many are the same and how many are not the same. So take 15 as the low end of the range. Now, we don't want that in research, and we don't want that in diagnostics, because nobody's going to try and muddle through 15,000 proteins. But at least feel the just sheer density and power of that lineup and what can come out of it, because they're going after everything that might underlie the detection of dementia and the treatment of dementia. We actually have found 35 companies that are currently developing precision blood-based biomarkers for dementia, neurology, Alzheimer's, and all the rare diseases associated with it. So, a word about GAP. When we got frustrated with our inability to detect people that had or didn't have the pathology characteristics that would make them good candidates for the therapeutics, we've started our own blood-based, well, actually our own biomarker study, Bio-Hermes 1, started in COVID. We had extra capacity at the time because of COVID, and finished it in 22 months, 1,000 people. We looked at CNs, cognitively normal, MCI, mild AD, and we tested a huge battery of both blood tests and digital cognitive tests for the specific purpose of trying to disassociate the commercial noise from what the hard data was in performance. that study turned out to be one of the 3 data sets that Fujiwara used to get its first clearance. And with that approach, we've become increasingly confident. It was look at May 16 of last year. So in the last year, that clearance has created another wave of both anticipated success, but also likelihood of bringing to market clearances in the FDA, from the FDA, that are going to give physicians and clinical trials working with participants a much higher level of credibility of what we're going to say to them in the course of their actually being put into trials or being diagnosed for disease. Now, this is for all of you who are trying to find rare disease. We actually published on this. So in Bio-Hermes, we had 600 people that were characterized as either MCI or mild to moderate AD by real top-notch PIs. Of that 600, a little less than 300, but we called it Mohs 300 because that's cooler than 296. The Mohs 300, who's our chief science officer, showed that of this nearly 300, there was no demonstration of amyloid positivity at all, both on PET and in the blood tests. We couldn't get a clear signal when we analyzed them from 3 different proteomics panels that we have in Bio-Hermes 1, as well as a full genome panel, and nothing was coming through, which speaks to the heterogeneity of dementia. But more importantly, we did find one signal. Across all of them, in both the positives and negatives, we had an NFL readout. Now, I've learned all this by hanging out with the scientists, but if we can show NFL, we know it's neurodegeneration, not likely something else that is driving their dementia. Now we have to ask ourselves, what is that? Multiple pathologies are at work. And that's what we're dedicating ourselves to drill down on that harder. Another thing came out of Bio-Hermes 1, which is we were able to show in the United States population, I in no way suggest it's true anywhere else, that when you think about dementia, I have a very profound history of Alzheimer's in my family. And if you looked at the Alzheimer's Association data or related advocacy groups, they would say of dementia, 70% of all dementias are Alzheimer's. So we did, because we had such a robust underrepresented population in Bio-Hermes 1, it was 24% non-white. And we were able to demonstrate that when you tested for amyloid, dementia positives were actually going to be about, in the Caucasian community, 50 to 60% amyloid positive. Then when you added our American Latino community, it was 35 to 40% amyloid positive. When you added the African American community, it was between 30 and 40% amyloid positive. So when you weight by incidence by race and ethnicity in the United States, there's only about 40% of the dementia cases are actually ATN Alzheimer's. The rest are something else. And we all have to dig in and find out what that 60%, not 30%, really represents. Oh, I'm going to back up, just last line. So we made all our data publicly available as of August 1 in Bio-Hermes 1. And we keep adding. We have a different way of doing things. So for example, in that data set now, you'll find the full Alamar blood test 4 years later on 220 individual tests against the 1,000 people we have in it. It's the first public expression of their technology in the Alzheimer's field. You're all welcome to call us. Happy to give it to you. We've had 140 downloads of the data set. So then we started Bio-Hermes 2 to dig into the Mohs 300. These are the partners. You might notice LifeArc there. But you can see that's a pretty wide range of little companies and big companies, huge pharma companies. And you would ask yourself, well, why are they all jumping into this and helping to underwrite this study? Because even in Roche or Lilly or Biogen, they can't get the money in the budget to run a trial like this. So they, rather than that, they buy a piece of the trial and get to share in the data. This is another 1,000 people. We've got tau PET, amyloid PET, a very, very demanding, particularly demanding MRI, and once again, even more blood tests, as you can see. Allow me to just point out a couple things. There's Spear Bio, Alamar, and Roche, Siemens, a large number of big diagnostic and small diagnostic players. Spear Bio wasn't even a twinkle in our eye in 2017. Neither was Alamar. It started in 2018. And they're bringing a next generation of blood test technology to the marketplace. In addition, thanks to LifeArc, we have the Capitainer blood spot technology being tested. And finally, a great, we didn't advertise it at first, a real emphasis on trying to find small vessel vascular disease. We're going to give you, as researchers, the most well-characterized data set we can conceive of to get a shot on goal for small vessel vascular disease. Very difficult. No one's beaten that. Nobody's put that in a blood test. Also, we're helping the alpha-synuclein, progranulin. We've got a long list of what we're going to try to find after we finish. So this study is now fully enrolled. It's here in the UK. We have, thanks to, I did want to put a word of thanks to Innovate UK. 4 UK companies are in this, AI Gnostics, Cumulus, I know I'm going to forget something, ILOF, it'll come to me, are all IUK beneficiaries. They funded their participation in this study, and we're going to be bringing their results through as we conclude the study for all of you to evaluate. So this is the slide. This is the slide you've never seen before, I bet. Anybody ever seen the Alamar public offering? So Alamar is what they call in finance the pure play. This part belongs in the blue room. But Alamar is a blood test technology, very small quantities needed, very precise, very critical to this is that the underwriters said, what is the growth in advanced proteomics and advanced proteomics estimated in 2035? 10-year growth factor. And so in that regard, they said, today it's $4.1 billion. That includes everything, cancer, everything else. But the point is, all the growth is not going to be in cancer because they've been pretty darn busy to begin with. So in the fine print, they evaluated that the growth in these kind of blood technologies would be a CAGR, compounded annual growth rate, of 37%. You've never seen that in neurology. And yet I think we're going to see an explosion of capital, explosion of technology that allows everyone in this room to be much more precise with the way we go about what we're doing. And if you have that kind of growth rate, we should all be optimistic we can get what we want. So I think I'm out of time. So I'm just going to finish the last bullet that matters most. If there are so many companies generating blood tests of such enormous precision, increasing precision and versatility, let's give them the data to do it. But it's incumbent upon the neurology field to really realize that these tools are coming. And we need to be quite prescriptive about what it is we want, both in terms of data and performance. And what are the targets we want to go after, because they are going to have the money and inclination to serve us if we are clear with what we need. Thank you.